and delegates. It gives me great pleasure to put my research in an international conference in Dallas. We are familiar with herbal medicinal plants, but we are not much aware about the standardization, because herbal medicinal plants, the routes of coming in the direct markets is a very, very long travel, because whether it is coming from cultivation or whether it is coming from the wild, nobody knows. But people are relying on the herbal medicine which is being sold in the market. If we consider about the facts, 35,000 medicinal plants are listed all over the world, and out of these 35,000 herbal medicines, 31,000 herbal medicines are present in India. Despite being the huge or hard use of synthetic medicine, still our Indian population is depending upon 50 to 70% on herbal medicines. 80 lakh registered medicinal practitioners of our Indian system of medicines, 7,000 registered medicine manufacturers, and 8,000 pharmacies still depends upon herbal medicines. In our Indian pharmacopoeia, 100 important medicinal plants are being listed, and 80% of these still they are mentioned in Indian Pharmaceutical Codex, that is British Pharmaceutical Codex. Herbal medicinal plants are not accepted in Western medicine systems because they have not yet standardized. But byproducts are accepted. But these byproducts are directly prepared from the medicines which has been purchased from the direct market, herbal market, or from the cultivation. But this problem arose in our mind, whether these medicinal herbs or medicinal plants are free from diseases or not. Because when they are in cultivation, different soil microorganisms are associated with them, endophytic fungi are associated with them, and when they are coming in the market after harvesting, whether they have properly dried or not, where their percentage of moisture is properly maintained or not, whether the transport system is clean or not, whether the storage system is up to date or not, whether they have not stored in the clean and dry container or not. These variety of questions are actually associated with this problem, and I have tried my level best to standardize some indigenous medicinal plants and some drugs, that is Dashmulaarist, that is 10 different medicinal plants are used in preparation of Dashmula. It is a very good tonic. And these are some of the herbal medicines are repeatedly used in herbal preparation and their byproducts. And what we did, actually, we studied the microflora associated with these plants. And these results are very, very different. And these are the methodologies we used. Collection of samples, various plant samples were collected, isolation of flora, that is microflora associated with these plants, with the blotter test, agar plate methods, then counting of fungal colonies on culture medias, identification of fungi, then effect of relative humidities. We maintain these herbal drugs under different relative humidities from 15 to 96, and we got different fungi under different percentage of the humidities, and the period for interval of 15 days we maintain here. And no doubt, being I am a pharmacognosy person, detection of therapeutically active principles from the drugs, that is my major field. These fungi are found to be growing or associated on these plants. They are nourishing on these plants. They are deteriorating the chemical constituents from these herbs. And these are we are consuming. But we don't know about how microflora we are consuming and less potent drugs we are consuming. This is our intention to try that. And we got the results. These fungi are actually consuming the most of the reducing sugar, non-reducing, that is starch containing proteins, phenols, alkaloid, glycosides, and all other chemicals which are present in this. Then after this, we did the phytochemistry, that quantitative analysis, then aflatoxin contamination, whether they are associated with this. And you will enjoy all these slides. You will see this blotter test method. We have inoculated on agar method, surface washing methods. We are scraping this surface and we are inoculating that. And this is the basal portion of this sclerodon, one of the plant. You see how much fungi are found on this sclerodon. Then this is around Culen colerifolia, that is one of the very good medicinal plant. You this fungi association on this. Large microflora is associated with this. Then occurrence of fungi associated with this method, these types of fungi characterized. Aspergillus species is a major species associated with herbal drugs. And these are the different Aspergillus flumigerus, Aspergillus terras, Aspergillus parasiticus, Niger, elegans, Oryzae, flavus, rubrum, globulum, spirelli, Chitomium species, Fusarium species, Trichompsis species, Rhizospum species. These are the fungi found to be associated on these herbal drugs. Then characterization and identification of these plants. These are the different Aspergillus species photographs identified. Then this is the Corularia species, Chitomium species, Chitomium 2 to 3 species, 4 species, Fusarium, Trichurus and Rhizospum species. Then this is the list of fungi associated with the studied drugs with their percentage incidence. 33% is the more percent of Aspergillus parasiticus. It is found in this plant. Here it is found in 19%. These are the different percentage found in the species. We calculate the percentage. Then in totality, what we got it here, Acorus calamus, vacant drug, it is a very potent drug. 10 species of fungi, 10 fungi associated. Boravia diffusa, it is again 13, Cassia 12, Scleroderm 9, Phygonia 10, and this one. These are the different. Then another method also used for supporting these results, that is the plate showing the herbal drugs stored under 96 humidity percent, relative humidity. You see this effect of relative humidities on different plants. These are associated with fungi, and these are their results in totality. under 33%, 55%, 75%, and 100%. These fungi were colonized on the herbal drugs. Then again, another plant species, Boroi diffusa. These are the results. Then another plant, Senna cassifolia, angustifolia, purgative drug. These are the percentage. Then another plant, that is the coloradon or seriatum. This is Cullen coryllifolia. This is Phagonia critica. Then these plants are having some chemicals. These chemicals are present or noted here. And they are showing their results diagrammatically with the help of pie diagrams. And they have shown their results. Humidity, different species. And it is noted that after 33% of humidity, usually fungi are invading on the drugs in totality of the result of these species, humidity percentage. Because there is a standard protocol for how much percentage of the moisture should be allowed in the drug. But if we are not standardizing it, they are crossing that limit. And ultimately, these fungi are associating on these herbal drugs. This result indicates the relative humidity experiment. Then deterioration of sugar percentage. It is actually huge data, but I have accumulated in with a very difficult. Then determination of sugars. Then this is aflatoxin tests. We followed this, but none of these drugs shown the aflatoxins incidence. Then what we got, the quality of studied herbal drugs is retained by storing them at lower humidity. This is around. It is also observed as 75, 96, 100% showed significant reduction in the sugar level, proteins level, phenols level, glycoside level, alkaloids level. Selectors are also screened for aflatoxin contamination, and it was observed that these drugs are free from aflatoxin contamination. Quality control to prevent growth of fungi and aflatoxin contamination in these drugs is essential. Hence, it is considered worthwhile to analyze some of the potential herbal drugs. It is found to be drugs stored in container, warehouses, maintenance, low temperature, low humidity level. So these are our suggestions. But it's a really interesting problem, because wherever the herbal medicines are used in all over the country, the worldwide survey or countrywide survey of such type of study is needed. And this is our beginning. It is not the end. Our research is still continued. And thanks for our authority of Pune University for financial support, my all research students and organizers for giving me an opportunity to present such type of work in an international conference held at Dallas. Thank you very much.